Thank you very much, everyone, for coming. We're a small group, but nevertheless, we're very enthusiastic. And thank you so much to Kew and to Paul Rees for having welcomed us here at the Kew Gardens at the nursery. We will be hearing a talk by Charles Lee a little bit in a minute, and then a few more slides from me introducing the species we're trying to resolve using DNA technique. And I hope you find this interesting, and I hope our members who will be watching this later will find this interesting as well. Without further ado, I'll hand the mic over to Charles. Thank you very much.
Okay, right. Good morning, everyone. My name is Charles. It's a great pleasure and honor to be giving a talk here in Kew Gardens. So a little bit about myself first. I did my undergraduate in the Department of Plant Science, University of Cambridge. Then I did my master's degrees in bioinformatics, University of Bath. Now I am doing a PhD co-supervised by University of Exeter and Dr. Ilya Leach, based in Drodger Laboratory here in Kew Gardens. I am fascinated by South African plants. Most of them are geophytes. I grow a lot of them, mostly from seeds. As you can see, Cathanthus, Stromaria, Getilis, Amaryllidaceae, and Conophytums, and of course, Section Horea, the geophytic clade of pelargoniums. So, on to the title of my talk, which is how to tell apart a carrot from a spinach. Well, when I told this sort of title to my supervisor back in Exeter, who knows nothing about pelargonium, he was like laughing because like everyone can apparently tell apart spinach from a carrot. You know, their spinach is with entire leaves, and you know, carrots with this very finely dissected leaves. And how could anyone not know what they are, right? However, those morphologies, usually very distinct among different plant groups, become very muddled up in pelargonium, especially in Section Horea. And I'm sure Matija is going to tell us more later. So I won't be I won't be divulging much, but to remember that this sort of leaf morphology is creating a great issue for taxonomists and botanists to resolve the species using this traditional morphology-based taxonomy. So a little overview of what I'm going to be talking. So overview of this Pelargonium genus, which I'm sure all of you will be very familiar with. The key morphology traits of Section Horea and why is it difficult for us to distinguish this species from a traditional point of view, and the outstanding species complexes. And then I'm going to talk a bit about how have the past researchers done on the phylogeny in genus Pelargonium, and finally how we are doing it in our project Pelargonium, funded by the IGG, our sequencing techniques and our tree building mechanisms. That was supposed to. So Pelargonium is the biggest genus in the family with almost 300 species. And it's a basal clade in the family as well. And as we all know, Pelargonium has long been introduced into horticulture ever since it's discovered. And now many cultivars exist, and it's known commercially, and I hate that, as geraniums, you might have heard of. Yeah, but they're not true geraniums, but we'll let it slide. Pelargonium obviously is very important, both in horticulture and economically, as a huge source of essential oils and creating millions of words per year. And within the genus Pelargonium, the largest section is Horea, which is surprisingly heavily underrepresented and understudied. So why is Pelargonium, especially this section Horea, so difficult and so confusing? One big issue is heterophily. That is, the leaves, they develop different morphologies throughout growth season and throughout time, or with different environments. So as you can see here, there are two pictures, both on Pelargonium dipetalum. As you can see here, the young leaf, the older leaf, the first emerged from the tubers, are entire. Then it becomes irregularly lobed, then deeply lobed, finely lobed, and essentially you could find some leaves by the end of the growth season to be finely dissected, even like, feather-like. And this great change, this drastic change in morphology is a great source of confusion. Just to imagine, if you are like a botanist and you are on an expedition, and if you were to collect this sample at the start of the growth season, compared to you're collecting it at the end of the growth season, you might as well just describe it as two completely different species, because herbarium samples are usually just a snapshot of what it looks like at a very specific time point, and that is usually lost in those samples. So one other thing I wanted to point out is I've seen a few people in the groups and other literature referring to this as heteroblasty. This is actually not heteroblasty. This is heterophily. The difference between these two is heteroblasty is a complete change, and usually irreversible. It's a complete change in gene expressions to change its entire morphology to as a sort of a complete shift from juvenile to mature, or from one state to another. So you can think of as like many forest understory plants in Madagascar, having different leaf shapes, but once they reach maturity, they will they will change their leaf shapes altogether and will not revert back. Or kind of like how locusts turn from non-swarming form to swarming forms, and these are heteroblasty compared to this reversible and periodic heterophily. This is just a little excursion on my yeah. So as I've talked about, there are species complexes in Horea, and quite difficult to resolve. So as far as I'm aware, there are 4 major complexes in the section. So the first complex, which is the one I have been working on this year, is this complex 1, this proliferum complex, which includes Caledonicum, Longiflorum, Longifolium. I'm not going to name all those. I will be showing you some more pictures later. Then complex 2 is this yellow-flowered complex, which yellow-flowered complex across Western Cape, essentially, and they are quite difficult to tell apart as well. Complex 3 is luteolum and gracillimum, and complex 4 is rapaceum, with its endless ecotypes, catering to a wide range of pollinators. So this year, IGG's grant has allowed me to sequence and to extract the DNAs and sequence most accessions in complex 1, and we're hoping to resolve that later this year. So a little bit more pictures that I found online about the proliferum complex. So proliferum kind of look like this, with this dainty little flowers with feather markings and those lanceolate leaves. It has an interesting sort of distribution, kind of like an L-shaped, across both Cedarburg and Overberg. And that includes 2 soil types as well. That, of course, I'm talking about, the sort of in in a very lax sense. Because who really knows which species is one another, right? Because this is literally what we're trying to find out, whether they are all the same or, whether they are splittable, right? So Pelargonium proliferum sensulato has this interesting L-shaped distribution, sort of from here to here. And if we're looking at eastern populations, they are usually like this, little rounded petals and less prominent nectar guides. This form has been sort of tentatively described as Transmontanum, named after the Overberg Mountains. If I may say, this sort of rounded pattern are almost like bridging some other neighboring species, like Montaguense and etc. Then if we're moving westwards, you can see again this very interesting heterophily here. The first leaf it puts out is entire, then it's lobed and more feathery, and by the end of the flowering season, you have this very finely dissected leaves. So this is very confusing and creating a lot.
Okay, so it's like why bother? Just no.
Yeah. They just couldn't make up their minds.
No, exactly.
Yeah. Yeah. So as we even move more westwards toward Cape Town, the morphology become even more. This is like almost like a Caledonicum. I found this on iNaturalist. I think this is a Caledonicum, but they are bridging the morphologies between Proliferum and Caledonicum, and previously that is bridging the morphology between Proliferum and Longifolium. And here, as we move very northwards onto the Cederberg Mountains, we find population like this with very tiny rosettes that don't really have dissected leaves at all. And those rosettes are also sticky. So a trait we call samophily. So they stick sands onto the surface of the leaves to protect from other abiotic stresses. So towards the northernmost end of the population, we find flowers with big blotches as well, and leaves very sticky, bridging the morphology with Samophyllum. And the flowers, I wanted to point out, with big blotches are usually a telltale sign of switching from butterfly/bee pollination towards beetle pollination. So all this range, different ranges of morphology, and all those populations being put on like a spectrum makes the delineation of species very difficult for us. And with more observations with more observations and more populations found by expeditions, as well as more data and pictures being provided by hobbyists of those populations in horticulture, more and more, the boundaries between the species has become more and more muddled and less clear. So it is important for us to really know that how those species have come about, and how have they sort of radiated and speciated, and finally whether they are just the same thing but just with different looks. So that is where we come in. We sequence the DNA, and based on the sequence data, we try to produce this evolutionary phylogeny tree between those species, and try to, and with that we are hopefully able to tell whether all those are the same species or they deserve to be split. But before our project, a lot of studies have been done on, a lot of phylogenetic studies have been done on the genus Pelargonium. Before 1993, there are some attempts to create this phylogeny tree, but mostly based on morphology. So it is a character-based cladistics search method to essentially build a tree. And and in year 1993 is the first subgeneric phylogeny produced with both morphology and karyotyping. So karyotyping is what you essentially extract all the chromosomes and you sort it by the chromosome morphology. So this is the very first DNA-assisted so this is the first phylogeny built based on DNA morphologies, essentially. Then in 1994, with the PhD thesis produced by Elizabeth Moraes, is the first and only section Horea monograph to date. And that's already been over 30 years now. In the thesis, there's no section-wide phylogeny, but she has grouped those species by their morphology. And the year came to 1995, so this first sequence-corrected phylogeny was produced, but was only in a very few species and not at all reflective of the entire evolutionary history as a whole. In year 1998, people started to sequence the plastid genes and started to build a purely DNA sequence-based phylogeny. However, they are only using very, very few markers. So Bakker from 1998, 1999, all the way to 2000, he has collected this mitochondria and chloroplast genes, 2 markers, on a range of species, but only 1 Horea species was included. That sort of that work has consolidated the place of section Horea within the genus. However, it does not include any sort of insight within the genus, the way we're trying to look at. And in 2012, there is another sort of great expansion in the scope of Pelargonium phylogeny research, and they've used 5 more plastic markers, and 8 Horea species were included. And we have since seen some problems with the phylogeny building with plastid markers, that is mitochondria and chloroplast, not nuclear sequences, because as you can see here, that was the phylogeny they produced. A lot of nesting was present. As you can see, Jenkinsonias are nesting here, and a few are nesting between Peristeros, and a few are nesting between Polyactiums. However, section Horea itself is very standalone, and we can pretty much be sure that Horea is a sort of phylogenetically sound group. So the widest generic revision was done in 2014 with 2 plastid markers, but for 110 taxa. And again, section Horea was heavily underrepresented. Out of the 110, only 6 species were included. Even though they have corrected the phylogeny, still it does not provide much insight on the evolution within this geophytic clade. And after all those years, in 2021, there's another study on section zirconium, and this is the first nuclear DNA-based level phylogeny. However, it does not apply to us because they specifically chose distinct species to begin with. Essentially, they're testing whether this phylogenetics works on zirconium. So they're already with a result in hand, essentially. So what we are doing in Project Pelargonium is, this is the phylogeny produced with the 2014 paper, with all the other sections being resolved. And as we can see here, Horea is being placed on top, meaning suggesting it's the most evolutionary advanced group, in a sense. But again, with only six species in the phylogeny, you can hardly tell anything. That applies to us. So, Project Pelargonium is supported by the IGG Small Grant, and I kindly received those leaf materials collected in Matija, and I've done the DNA extraction in Georgia Laboratory, just over there-ish. And then I will be receiving the sequencing data, then I will be carrying out those bioinformatics work in University of Exeter. So this was me 2 months ago. I was weighing out the samples. As you can see, pretty much the leaves are looking drastically different, from entire to this irregularly lobed to the finely dissected to like feathery. So the first step is to weigh them out, then put into this big grinding sort of machines to essentially pulverize it. Then those leaf powders can be used to extract the DNAs. So let me tell you a bit more on the sequencing strategy we're choosing. So instead of using only 1 or 2 markers or looking at those plastid sequences that are not within the nuclear genome, we're using this angiosperm 353 kit. Essentially, it is a series of probes for capturing 353 genes. So this 353 single copy protein coding genes are selected, manually selected from this 1,000 transcriptome consortium, essentially to provide a overall sort of landscape for all the universal nuclear genes in flowering plants. So instead of getting only sequences from 1 or 2 markers, we'll be essentially getting 353. And it's capturing both exons and introns as well. So with better resolution, we can essentially do more work to tell apart even intraspecific relations, such as different populations of Pelargonium proliferum, and whether any particular accession is worth splitting or not. So with angiosperm 353, we're hoping to retrieve that many genes, but as with science and with life, it's usually not going to be like that. But it is a very robust system, and allowing us at least to extract 150 loci. That is, we are at least going to build 150 trees and combining their topology to give us this consensus tree and to consolidate their evolutionary path, evolutionary relationships to one another. What else am I going to say on angiosperm 353? Pretty much that's it. Oh yes, it has also been used in other species level phylogenies, such as primroses, primula, and pitcher plants, Nepenthes. So we have already sort of known that it has it has been known to be effective on those species level resolutions. So a little overview of the timeline. Last July, I sent this proposal to Matija, and I've quickly gotten the grant. And by October last year, we have started to settle on the species we're looking into and preparing leaf samples for drying and storage. And then by February this year, I received the samples. And May this year, I was here to do the extraction. I don't know if you're in the Facebook group. I did a few posts on that. And just this last week, those samples have been sent off to sequencing. And hopefully by the next 2 weeks, I'll be able to receive the data and start working on the phylogeny. And hopefully by the end of this year, we'll have this first draft of phylogeny. It's going to take a bit more time compared to those previous studies, because they were looking at just 1-2 locus or loci. Instead, here we're looking at 353. So we're building essentially hundreds more trees and trying to resolve them. So please be patient with me. So to conclude, why is this work relevant and important? Pelargonium is firstly important both in horticulture and economically. Section Horea is the least phylogenetically studied section, even though it is the biggest section within the genus. Now it's pushing 90 species now, I think. It was 85 when I wrote the proposal, but then like a few more came out. Yeah. It has been more than 10 years since the last phylogeny was done on the whole genus, and it's more than 30 years since the last sectional revision. So with this many new species being produced, described, and being put out, it is timely for us to do this work and resolve some of those species complexes. I want to say thank you for everyone that came here, everyone else listening online. If you're my mom, go to bed. I don't know. Yeah, and thank Matija for collecting the leaf samples, Benjamin for sorting out the finances, my supervisor Ila Leach here in Drodrell, who hosted me during the DNA extraction, and my lab group here back in Exeter. So yeah, that's pretty much it. Thank you for listening.
Okay, thank you very much, Charles, for your presentation. I'm afraid that my level of knowledge of genetics or botany even is so much lower. I'm just going to show you a few nice pictures and tell you a little bit more about my experience with pelargoniums in habitat and in the greenhouse. But I will talk about the same complex of species and even show a few of the same photographs just to drive home the point of how difficult it is to identify these species either in nature or in the greenhouse. Very difficult in the nature because it's very often Horeas flower in the spring, summer or autumn, whereas they are in leaf during the winter. So they very often flower and the leaves are not there. And for identification of these species, you evidently need both. And I think that a lot of the early identification work was a little bit flawed simply because either people relied on flowers a little bit too much and they didn't recognize the differences in leaves, or the other way around. Because you've got species that have almost identical leaves but vastly different flowers and vice versa. And that has led to a lot of confusion. And Pelargonium proliferum and the related species that we're trying to resolve with Charles are one of those species where you've got a particular flower type and a lot of very different, not just leaf shapes, but also leaf hairiness, if like. So I'm going to focus a little bit on Pelargonium proliferum first. This is a species that can be easily identified by the short nectar tubes, which are typically up to about 15 millimeters, 10 millimeters more likely, and very short petals with prominent nectar guides, but the petals are about as long as the nectar tubes. So they're also short and they are typically linear, so not spoon-shaped, if you like. So that's the typical flower. It's repeated in several taxa, and that has led to a lot of confusion, I think. Because the same flower type is shared by some other taxa, which we believe are different species, I'm going to show a few of them. One of them was found by Rian van der Walt, who's actually the son of the Professor van der Walt, who studied Pelargonium in the 80s and in the 90s. And Rian found the species in a very nice nature reserve close to Cape Town, one of the few Renosterveld patches that still exist in the Swartland, which is a heavily, it's a vast agricultural area. And in these patches, a species, a taxon grows that, as I said, flowers very similarly to Proliferum, but the leaves are super, super different. This is basically the area that we're talking about. In iNaturalist, this is the distribution of Proliferum as a complex, as a whole. We're going to look at a few specific populations, which I think are quite different. The Overberg populations are, I believe, a separate taxon, but Charles will tell us if that's right. At least morphologically, they look quite distinct. The populations spanning across the Cederbergs and over to the Overberg is what I think the true Proliferum species. Then the Swartland and closer and slightly more southern species are southern populations are, I think, a separate taxon. And there's a there's a taxon with a very, very small distribution around Darling, which also appears to be quite different. But let us have a closer look at those. Rian's species, which provisionally we call Pelargonium riani, has almost glabrous leaves, so not hairy at all, although under a microscope it does have very short hairs. But what's really, really important for this particular taxon is that the leaves have petioles that are bent, and that's very often seen as an important character in the identification of Horaeus. And the other really important character of this species, you see that the flowers are exactly the same as of the proper Proliferum I've shown before. The other important character is that they flower in the autumn. Proliferum proper flowers in the spring and into the summer, whereas this species flowers several months later, so there's probably enough of a difference in the flowering time to separate it from Proliferum proper. This has been morphologically studied. A herbarium specimen has been deposited at Kirstenbosch, and we're waiting for the DNA results to confirm or not whether, whether in addition to morphology, the species or the taxon could be separated on the basis of DNA. The Overberg species or the Overberg taxon is quite a bit more widely distributed because although only about 5% of Overberg Renosterfeld remains, the taxon is quite widespread. It's quite numerous in the patches where it grows. This is a very nice and hilly area, very developed agriculturally as well. The species is extremely small. It has, as opposed to the other, to the true Proliferum, it has very, very long hairs, which are not a character of Pelargonium proliferum proper. And if it grows in the open, it develops very small rosettes of about 3 centimeters across, an inch across. But if it grows in between grasses, it develops quite tall leaves, and then those leaves tend to be pinnate. And that confirms perhaps my observations, my greenhouse observations, that if the light levels are low, the leaves tend to be pinnate, maybe. So I think we need a little bit, a few more observations. The flowers are also quite different because they're spoon-shaped or spatulate. They tend to be, the petals tend to be a little bit larger than Proliferum proper. I'd be very surprised if we didn't find this as a separate taxon in DNA. As I said, it's very widespread, and it's currently identified as Proliferum. It also flowers later in the summer, so quite a bit later. Mine are not flowering yet, and it's July. They usually tend to flower in August and into early September in the northern hemisphere, of course. The third taxon in this complex is a taxon that grows in very numerous populations, but has been only spotted around Darling so far. It has mostly entire leaves, mostly glabrous, although it does have short hairs along the leaf margins. But what's really interesting is that it has very, very long and papery stipules, and that is not a typical character for Pelargonium proliferum. The leaves are mostly entire, although some leaves tend to be lobed as well. And as you can see, the petioles are not bent, as we've seen in Pelargonium reani. So this tends to be a little bit different when we look at the leaf shape. And of course, when we look at the flowers, it's exactly the same as they all are. So quite prominent nectar guides, very short nectar tubes, and linear petals. This also flowers very late in September in my greenhouse, so it can be easily separated from Proliferum proper, which flowers in spring and in early summer. So again, I'd be very surprised if this isn't nicely separated in our DNA studies as well. Psammophyllum is another really interesting species, described fairly late, when it was separated from Pelargonium longifolium. It also has variously shaped leaves, as nicely described by Charles. Very often the leaves, this is a population on the Cape Peninsula. Very often the leaves are much more finely dissected than that. And the flowers have very broad posterior petals with prominent nectar guides, and they tend to have an intensive blotch, but this population didn't have blotches, and according to the description of Elizabeth Murray, that's okay in terms of the species description. However, the species as a whole has quite a broad distribution, and we now know that the populations from the Cape Peninsula up into the southern Cederbergs and into the western Oberberg, these populations all tend to have pink petals and quite finely dissected leaves. As we move further east, the populations around south of Bredasdorp tend to have white petals and incredibly broad. I'll show a few pictures later. And these might turn out to be quite separate. As we move north from Cape Town, the pink flowering populations turn to yellow flowering populations. It's very odd for a Pelargonium species to transition from pink to yellow. There are a few cases, such as Gracillimum and Luteolum, for example. Also, Estivali can flower pink or yellow, for example, but these cases are not very numerous. And then, as we move even further north, in the Piketberg, the flowers turn to white again. So, at the extremes, we have white flowering, Psammophyllum, yellow on the west coast, and pink elsewhere. I don't think I personally don't think that petal color is a very important diagnostic character, but we'll look at the other characters as well. So this is a yellow flowering population on the west coast. We see the typical finely divided leaves, and they are much less hairy than those on the peninsula. But as I said earlier, I don't think that the petal color itself is a good character to separate two taxa. I think this is probably going to turn out to be slightly different, but not a different taxon altogether. As we move further north to the Piketbergs, this is a population just north of the Piketberg range. We see very small plants, rosettes barely beyond an inch, so 3-4 centimeters, and leaves that are pinnate. In terms of pinnation, this is not unusual for Psammophyllum, but the petal shape is very different. The petal shape in the Piketberg is very reminiscent of species like Naviculifolium or other similar species. So this might well turn out to be a little bit different. But currently, it's identified in herbaria as Psammophyllum. In the eastern Overbergs, we get species with very different pinnation. So we get bipinnate and tripinnate leaf shape, and very dense hairiness. And these are the populations that have extremely broad petals and very often very long nectar tubes. The nectar tube is a very important character in pelargoniums because if the nectar tube is longer than about 20 millimeters, it's understood that the species cannot be populated by bees because their proboscis are not long enough. So, and these nectar tubes are typically around 20 to 25, sometimes 30 millimeters, and this would indicate a little bit of separation from other Psammophyllum populations. In the southern Cederbergs, we get populations with very finely divided leaves, so very similar to Cape Peninsula, and petal shapes that are much more typical for the species as it has been described by Murray. There are other populations that I cannot make sense of. This is a Brede River Valley population in the same habitat as Pelargonium uliginosum. So this is an extremely special habitat. It's an alluvial plain that's often underwater, so flooded by the river. The leaves are mostly entire. They're mostly hairless, and the petal shape, I don't really know what to make of it. These are not the broad petals typical of Psammophyllum. If I only saw the flowers, I'd say longifolium, but I really don't know how to identify this particular population. So let's hope that the DNA work will tell us a little bit more. The petals are obviously linear and much longer than Psammophyllum, so I guess this could be longifolium as well. But it lacks the typical longifolium blotches. Another really interesting complex and very related to Proliferum is the complex of Pilocellifolium species. The typical Pilocellifolium that we know from the Overberg has glaucous leaves, so slightly waxy, and it has very thick hairs or bristles along the leaf margin, so that would be quite typical for the taxon, in addition to the petals having extremely strong coloration, extremely strong blotches on all 5 petals. The nectar tubes are short, and the inflorescences tend to be in the shape of a wheel. So looking at an inflorescence from the top, the flowers would look as if they are arranged as in a wheel. So that's the typical taxon. That's also described in Betty Murray's PhD. But there are many other probably closely related taxa growing elsewhere. This is a Kamisberg population, which is a fairly recent discovery. We now know that that the taxon, or that the populations with these leaves, which are very finely pinnate, they are almost indistinguishable from grass. They can be found in a few populations across the Kamisbergs, but the flower shape is exactly the same. Then we have populations close to Calvinia, which also have glaucous leaves, but they typically lack the bristles that are typical for this species. And then in the Cederbergs and in the Picketberg, we get species with very finely pinnate leaves, lacking the glaucous leaf surface, and lacking the bristles along the leaf margins. So this might again turn out to be quite a distinct group of Pilosellifolium populations. So personally, I think we might find that there are three groups of taxa within the Pilosellifolium taxon or species: the Kamisberg population, the glaucous leaf population extending from Calvinia to the Overberg, and the Cederberg and Picketberg population. And this is where they can be found. So the glaucous leaf Pilosellifolium extending from the almost eastern Cape all the way up to Calvinia. The Cederberg populations with very finely split leaves, very finely pinnate leaves, and the Picketberg populations, and the Kamisberg populations, which obviously are somewhere here but are not noted in iNaturalist yet. And that's it from me. This was from a trip with Rian van der Walt, when on a very cold winter day we were braving the rain and looking for Pelargonium heterophyllum. Thank you very much.